Hello friends, today we are going to discuss about transduction in bacteria. So first of all, what is transduction? Transduction refers to the transfer of a part of DNA between two bacterium, that is donor bacterium to recipient bacteria via a viral vector. Here the viral vector is bacteriophage. So in this process, with the help of the viral vector, fragment of DNA is transferred from one bacterium to the other bacterium, which are known as donor and recipient respectively. It was discovered by Zinder and Lederberg. Let us see general steps of transduction in brief. We have already discussed it in detail in transduction video related to NEET exam. If you want to know it in detail, you can click the link given in the description box. So bacteriophage virus infects bacterial cell, that is the donor bacteria. Using the bacterial machinery or the host machinery, it multiplies. And once the process is complete, viral particles are released. When those viral particles which carry bacterial DNA, either only bacterial DNA fragment or part of bacterial DNA fragment along with the phage genome, when these infects another bacteria known as recipient, then the genetic material is injected into the recipient bacteria. Here what we see that fragment of bacterial DNA is transferred from donor bacteria to recipient bacteria with the help of bacteriophage virus. This genetic material either integrates or remain extra chromosomal in the recipient bacterial cell. So transduction is a method of horizontal gene transfer because here genetic material is transferred from donor bacteria to the recipient bacteria, not from the parent to the offspring. Also in this process, physical contact is not required between donor and recipient. That is, these do not come in physical contact with each other, but genetic material is transferred from donor to recipient with the help of bacteriophage virus. And since the DNA fragment is present inside the viral coat, hence these are DNase resistant. DNase are enzymes which degrade DNA. It occurs through lytic or lysogenic cycle of bacteriophages. This we have already discussed in transduction video related to NEET exam. You can refer my video, the link is given in the description box. In this video, we will discuss lytic and lysogenic cycle in brief. Lytic cycle occurs by virulent phase. Bacteriophage virus infects bacterial cell and injects its genetic material. Then it starts multiplying using the host machinery. Once viral particles are packed, then these viral particles are released by the lysis of the bacterial cell. This is known as lytic cycle. Lysogenic cycle, it occurs by the temperate phase. In this, the bacteriophage infects the bacterial cell and injects its genetic material. Then it integrates with the bacterial genome and remain intact for many generations and replicate along with the bacterial genome. And when any induction occurs like exposure to certain chemicals or ultraviolet rays, then it is excised and enters the lytic cycle and the similar process takes place. This is known as the lysogenic cycle. So there are two types of transduction: generalized transduction, in short GT, and specialized transduction, in short ST. In this video, we will discuss about generalized transduction in detail. In the next part of transduction video, we will discuss about specialized transduction. First of all, let us see the steps involved in generalized transduction. Generalized transduction takes place by lytic cycle in virulent phase. First of all, bacteriophage virus infects bacteria, that is the donor bacteria, and takes the host machinery or the bacterial machinery for reproduction. Also, bacterial genome divides into fragments. Once multiplication or reproduction is complete, then the packaging of virus particles takes place. In this packaging process, few bacterial genome fragments are also packed by mistake, and this packaging is randomly. When fast particles carrying the bacterial DNA fragment, that is the donor bacterial DNA fragment, infects another bacteria which we call recipient, then the donor bacterial fragment is inserted or injected into recipient bacteria, and once it enters into the recipient bacteria, it has different fates. Either recombination takes place if it integrates into the bacterial genome, or it can remain extra chromosomal, or certain other fate is also possible. These we will discuss in detail in later part of this video. So let us see this with the help of diagram. Bacteriophage virus infects donor bacteria and injects its genetic material. Then using the machinery of the donor bacteria, it starts multiplying. Also, bacterial DNA or genome is fragmented. Once the multiplication is over, packaging of viral particle takes place. Sometimes, due to some mistake or due to some error, these bacterial DNA fragments are packed in the viral particle. Once this process is complete, viral particles are released by the lysis of bacterial cell. Viral particles carrying the donor bacterial fragment, when infects another bacteria known as the recipient bacteria, then it injects its genetic material into the recipient bacteria. So here, with the help of bacteriophage or via bacteriophage viruses, bacterial DNA fragment is transferred from the donor bacteria to the recipient bacteria. Then there are different fates of this bacterial DNA fragment. Either it remains extra chromosomal, or it is integrated and recombinants are formed, or some other fates are also possible. This we will discuss in detail later. Now let us discuss some of the facts related to generalized transduction. First of all, why it is called generalized transduction? Because in this process, any bacterial gene is transferred. That is, transfer of bacterial genome fragment is random. It is not specific. The probability of being transferred is same for each part of bacterial genome. That means each part of bacterial genome has the same probability of being transferred from donor to recipient bacteria. Different set of closely linked genes are carried by each transducing phase. Each transducing phase carry different bacterial DNA fragment or bacterial genome fragment. Typically carries only bacterial DNA, no viral DNA, and these type of transducing phase carries only bacterial DNA but no viral DNA. Generalized transduction occurs during the lytic cycle of virulent phases. This we have already discussed. A fragment of bacterial genome that is roughly the same size as the phase genome is mistakenly packaged during generalized transduction. So during the generalized transduction, bacterial genome that is packed in the phase particle is almost the same size as that of phase genome. Any bacterial genome fragment that is bigger than this can't be packed. Also, after completion of the lytic cycle, new virus particles, which is loaded with only bacterial DNA fragment, when released, infect another bacterial cell. So as we have discussed earlier, viral particles that are packed with bacterial DNA fragment, which is DNA fragment of donor bacteria, when it is released after the lytic cycle, these infect the another bacterial cell that is known as the recipient bacterial cell. So once viral particles that is packed with the fragment of donor bacterial DNA, when it is released, it infects the new bacterial cell known as the recipient bacteria and injects the bacterial DNA fragment. This type of the transfer is known as generalized transduction. So the bacterial DNA or the donor bacterial DNA fragment which is inserted into the recipient cell by the bacteriophage virus has three fates. First is it is degraded. Donor DNA fragment after being injected gets fragmented or degraded. It is then absorbed by the cell and recycled for spare parts. This type of transduction is known as unsuccessful generalized transduction. In second fate, if injected DNA was originally plasmid, it will recirculate inside the recipient cell and become a plasmid again. So, it remains extra chromosomal in the recipient cell, hence recombinant is not formed. And one or more donor genes that is present in this plasmid are transiently expressed. This type of transduction is known as abortive generalized transduction. No replication of donor DNA fragment takes place because it remains extra chromosomal. It does not integrate the recipient bacterial genome. Hence, it do not replicate along with the recipient bacterial genome. As a result of which, among the progeny, only one bacterium contains the donor DNA fragment. In the third fate, site-specific recombination takes place if the new DNA is homologous to the region of recipient chromosome. So, if the donor DNA fragments share homology with any region of the recipient genome, then site-specific recombination takes place. Hence, recombinants are produced and these are stable and are expressed. That means the donor genes that are present in the donor DNA fragment are expressed. These replicate along with the recipient bacterial genome since these are integrated. This type of transduction is known as complete transduction or successful transduction. Now let us understand these three fates with the help of diagram. So this is the recipient bacteria, this is recipient bacterial genome. This is the donor DNA fragment which bacteriophage has injected during the infection. So it has three fates. In one of the fate, this donor DNA fragment is degraded, then it is absorbed by the cell and used as the spare part for the synthesis of any genetic material. This is known as the unsuccessful transduction. In the second fate, what happens? If this donor DNA fragment is originally plasmid, then it recirculates. It forms plasmid and it remains extra chromosomal. That is, it does not integrate with the recipient bacterial genome, as a result of which it do not replicate along with this bacterial genome and the genes that are present in it are expressed for short period. Recombinant is not produced and among the bacterial progeny, only one progeny has this donor DNA fragment or plasmid. This is known as abortive transduction. In third fate, if this donor DNA fragment shares homology with certain region of this recipient bacterial genome, then site-specific recombination takes place. Now what happens? Recombinant is produced. So this donor DNA fragment is integrated in the recipient bacterial genome, recombinant is produced, this is stable and it replicates along with this bacterial genome. All the genes are also inherited. So recombinant is produced as this donor DNA fragment is integrated in this bacterial genome and it is stable. It is inherited because it replicates along with this recipient bacterial genome and the genes that are present in this donor DNA fragment, or in other words donor genes, are expressed. This is known as complete transduction or successful transduction. Frequency of abortive transduction is 1 or 2 orders of magnitude greater than complete transduction. Or in other words, abortive transduction occurs more frequently compared to complete transduction. Hence, what we can conclude? That most cells infected by generalized transducing phases do not produce recombinants. So this is all for today's video. In the next part of this video, we will discuss about specialized transduction. So stay tuned. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.